I'm Shelby. I am in the microbiology for health sciences lab, and today we'll be going over what a pure culture is and how to obtain it using the streak plate method. So a pure culture is a pure isolation of a single type of organism, and we can obtain this using several different methods like the spread plate method, the pour plate method, and as we know, the streak plate method, which we'll be going over. So the way we use these different techniques is that we take a mixed culture and spread it along a medium, and it slowly dilutes, thus obtaining the pure culture. Before we begin to actually streak the plate, it's important to label it as well. So on the back of the plate, we would write your initials, the date, as well as the culture you're using, but you would put it on the edges just not to obscure the view. After that's done, then we can begin streaking the plate. First step would be to heat the Bunsen burner up. Once that's heated, we can use it to sterilize our loop. So once we sterilize our loop, then we can dip that loop into the bacterial culture that we're using. So once that's dipped in, we can begin streaking the plate. You just pull gently through the plate, creating just like a zigzag type of line, and that would be only on 1/4 of the plate to begin. After that's done, we would sterilize the loop again and then repeat the steps, this time rotating the plate 90 degrees and 1/4 again of the plate, going over, gently just dragging it through, creating these zigzags, but this time we're going to cross over once into the other zone, the first zone we created. Now we created a second zone. So once the second zone is created again, we would heat the Bunsen burner, the loop through the Bunsen burner, rotate the plate again, 90 degrees, and then once that's done, we'll begin streaking again, repeating the process of dragging the loops gently through the medium. Once that's done, you would also as well drag it again through the previous streak that we just made, so through the second. So now we created a third zone. So once the third zone's done, we repeat the steps again. We would heat up our loop into the Bunsen burner, sterilizing it. Then we would rotate the plate 90 degrees again and gently drag the loop through the medium, creating a fourth zone. And this fourth zone as well would also one time be dragged through the previous zone, so the third zone. Once that's obtained, then you have your streak plate done, and then you would incubate it typically for 24 hours. Alright, so after we incubate for 24 hours, we should be able to see the results, which would be a pure culture of that organism, and we should be able to see the little dots on the plate, which would be those colonies of that pure culture. And it's really important for us to make a pure culture from a clinical specimen like this, because it allows us to observe and study the organism that is causing these issues on that individual, of that patient, and be able to create strategies based on that organism and figure out what the best method to treat this disease is. And the next step in order to treat the patient would be creating a specific treatment plan for them that would kill off that organism that is causing these issues that they're having, and typically that would be like antibiotics. So